good morning to everyone. Yeah, definitely good morning to everyone. And also, I feel really excited to see a lot of friends and also collaborators and even like the mentors in the Zoom chat. And I believe we introduced to everyone. I originally worked with the Chryptic project during my postdoc, primarily working with Alice Rogers and also Shawn Serbin from Brookhaven National Lab, and also work closely with Nate. So I saw a lot of like great friends here, and also today, and I have been enjoying Hong Kong U for since 2019, and so it's almost now my 2.5 year at Hong Kong U. So today I primarily try to start from what I have did in the past, and also how it linked to some like remote sensing approach my lab has been developing during the past 2.5 years. And I hope this can provide some insight to the modeler as well, especially like Professor Jianyang Xia mentioned about a lot about the model trait uncertainty. So we hope the remote sensing technology, especially the cutting edge remote sensing technology, can provide a way to advance this kind of field. So probably we can start from here now. So as we know, global warming now it's already become a really major issues and nowadays in our societies, no matter for its impact on our human society and also for the natural ecosystem. And also increasing consensus now has already been made is human immediate CO2 is the dominant cause of the global warming. And like the panel shown here on the lab panels, it summarizes two major sources of the anthropogenic CO2 emission, including fossil fuel consumption and also land use change. At least human immediate CO2 will go to somewhere, and about 44% goes to the atmosphere, causing the global warming, and remaining either absorbed by the land and ocean. And so that's also a reason why we, as ocean scientists, we try our best, try to help to understand and even to model land and ocean carbon uptake, because it's really critical for us to predict the future global warming and also climate change and also climate change impact. And I define myself as a land ecologist, so I focus a lot on my research on land components. And like Jing point out, if we now using an Ocean model to try to project the future land carbon uptake, actually there's a large uncertainty, and this large uncertainty can even higher than the current days, like the annual human immediate CO2 emission across the global. And one thing I want to highlight here is like the right panels summarize the Bonner and Donny work, try to quantify where is the sources of the uncertainty. And what they found are really intriguing is around 80% of the model uncertainty actually are associated with model processor and also model parameters. In other words, if we can improve our model representation of important ecological processes and also provide a novel way to parameterize the Ocean models, it will largely reduce the current Ocean models to project the land carbon uptake. So what kind of model processes we need to care? I think a lot, like Jing pointed out, but here I try to primarily use plant photosynthesis as an example I'll show here. And based on the slide here, probably we can also tell one important thing here is a lot of uncertainty probably is associated with the scaling processes, right? The nature is so compressed, and there's a lot of like scaling complexity that will induce the uncertainty. For example, take photosynthesis again as an example. The scaling complexity can be scaled from leaf level to canopy level, and even to even larger and spatial and temporal scale, right? So like a leaf level, for example, nowadays, if we already knows about leaf physiological trait together with some environmental drivers, then we can reasonably good to simulate the photosynthesis. And when we scale up to the canopy level, then in addition to leaf levels trait informations, then canopy structure become also important. But now if we want to extend this kind of simple concept to even larger spatial and temporal scale, for example, like the seasonal scale, then we found that we need to account for the phenology effect. But if we extend it further to the like ecosystem scale and even to the global scale, then we found that the plant function type will be really important concept. And even underneath of the plant function type is because of the trait, there's so many trait and also canopy structure can vary globally, right? And for today's talk, I will primarily focus on the phenology part. And when we talk about phenology, this concept probably is already quite clear to the audience here. But one likely example show on the left panel here is a typical like a phenology example in temperate decidual trees, right? It will show some like flush new leaf in the spring, then peak in the summer, become senescence in the autumn, and then drop leaf in the winter. But even use this single example, it will inspire us to think about there actually are two aspects of leaf phenology, at least two aspects of leaf phenology we should consider. And one part primarily associated with the canopy structure. For example, we can use the leaf area index as an indicator of the canopy structure, and that actually can help us to describe the quantity aspect of leaf phenology. And normally we can tell, even use the example show on that panel, we can tell the quantity aspect also shows really strong seasonal variation. At the same time, along with the seasonal development, especially with the associated with leaf aging processes, and we can tell leaf morphology, leaf biochemistry, and even leaf physiological trait also shows strong seasonal variation. And normally we can say this is associated with the seasonal variation in leaf biochemistry and the physiological trait, or alternatively, we can also say probably is a quality aspect. So why we should care about these two aspects? So the next, I would like to give you some example. Probably we can start from temperate decidual forest in the United States first. Like the phenocane on the top panels, it record there's a pattern, a leaf like the seasonal variability in this forest site. And in the bottom panel, the red color indicate is a leaf area index derived from the models. And the black dot indicate is the added covariance derived like the photosynthesis rate, like the short for GPP. And overall, we can tell the leaf area index seasonality can track on the GP seasonality quite well in this forest site. Now, if we move on to even multiple more sites, and we found a similar pattern actually holds up. But one intriguing thing is now when we move on to the spatial scale, it will emerge. So for example, now if we just focus on like the peak growing season, a maximum photosynthesis rate, and we found in the top panel, the GP, the maximum photosynthesis in the peak growing season is around like 80 grain carbon per square meter per day, which is much higher than the bottom panels. That means in addition to the phenology that can be characterized by leaf area index, some like the spatial variation of physiological trait are also quite important. And like the empirical observation shown here, and I found Zhen Yang and also his colleague did some really nice analysis in the 2015. So the main idea is they try to understanding about the linkage between phenology and also annual GP at the large spatial scale. And here shows the diagram here shows the seasonal cycles of the GDP. And then we can sum up all the gray areas as indicators of the annual GDP. And the basic hypothesis here is whether we can use a green triangle shown here to approximate the annual GDP, like the green triangles areas, whether it can approximate the gray areas shown in the figures. And we know in the green triangles, there are two key biologic factors. One is the growing season length, and the other one is the maximum photosynthetic capacity, and which is the indicator as an equal season scale physiological capacity. To try to test this simple concept, Zhen Yang assembled the added covariance tower data over a large across the entire American Flash network data, and they found even just using these two simple parameters, it can model the annual GP quite well across no matter across different forest site or the same site across different year. So that's quite intriguing. Suggests definitely the phenology of the quantity and also the physiological trait are so important to determine the photosynthesis of the annual cycle. So now we move on from the temperate forest to the evergreen forest, for example, like the tropical region. The story, whether we hold up, we are not quite sure. So, but one thing we can start from here is when I started my PhD in Arizona, and we collaborate with some scientists in the Amazonian in Brazil. So we try to install a phenocane in the Amazon rainforest to monitor the seasonal cycle of the landscape. And like the figure shown here, the top panel shows the August in 2013, and then until progress to the next year, like June of 2014. And we can tell across the entire annual cycle, when we see this forest site, it seems the whole forest maintain evergreen all year long, and it's really difficult to see if there's some significant change. But one thing I'm really intriguing is if we zoom in to see each individual tree crown, and we found there's some big variation. For example, like the top right panel, it just shows the one tree crowns, they become senescence in September, and then drop the leaf in October, and refresh leaf again in November. And when we move on to the next tree crown, and we found they drop the leaf in July, they flush leaf in August. And even some trees, they didn't drop the leaf, but will flush a new leaf in the next month. And when we carefully survey along 300 trees, three individual crowns across the entire landscape, and we found around 70% of tree crown will show significant seasonal variation in the leaf phenology. So that will raise another interesting question here is whether such individual scale phenology dynamic matters or not, especially in terms of carbon cycles. So to do that, we first developed some algorithm to convert the camera detect phenology data to the ecosystem scale leaf area index, like the canopy leaf area index shown here. At the same time, we can break the leaf area index at the ecosystem scale to different age cohort, like young, mature, and old 3 age classes. So like we expect tropical evergreen forests, right? They are evergreen. So leaf area index shows really small seasonal variation. But when we move on to the age cohort, and we found there are really large variation of the leaf age dynamic, especially in the dry season. And when we incorporate this kind of age information into the model, and we found the model actually can improve the photosynthesis seasonality simulations, like the red color shown here is a model with the account for the leaf age effect. The underlying mechanism is primarily because leaf behaves very similar like human beings. Their metabolic rate, approximate like the photosynthetic capacity, shows a strong age dependent with a really low values at a young leaf, then peak in the mature leaf, then decline again. So based on this, I would like to use both the temperate region and also tropical region as an example to show here is it seems when we talk about the two aspect leaf phenology, it seems are quite generalizable and also quite common for both in temperate forests and also the tropical evergreen forests. And the two aspects we highlight here, one is about the quantity, like a leaf quantity perspective, like approximate by leaf area index. And the other one is about the quality part, can be approximate by leaf level photosynthetic capacity. And in the tropical region, like we just mentioned before, this kind of like the age effect is primarily emerging from the diversity of the fine scale leaf phenology variability. And because of this, it motivates me to ask another scientific question here is, if fine scale variability in phenology of leaf quality and quality are so important for us to understanding larger scale ecological processes, and how can we monitor it? And in the next part, I would primarily like to suggest we have two potential solutions that correspond to remote sensing technique my lab has been working on during the past 2 years. The first technique we developed is called the high resolution select remote sensing technique that can enable us for fine scale plant phenology monitoring. And the second approach is primarily working with my colleague Alice Roger and also Shawn Serbin at Brookhaven, talk about the vegetation spectroscopy approach to help characterize temporal and spatial variability in plant functional trait. And in terms of the plant phenology monitoring, there's a really famous observation dilemmas in this field. On the left panel, it summarizes the conventional approach we can rely on ground proximate remote sensing technique for phenology monitoring. And this kind of technique, their advantage is for organism levels phenology monitoring with more accurate phenology records, but normally they are limited to the very small spatial coverage. Alternatively, we can also turn to the satellite remote sensing. They can help us to monitor at a global scale But normally, because they have a really much coarser spatial resolution, and normally they are more constrained at the ecosystem scale rather than at the fine scale. So the dilemma here is whether we can develop a novel technology that can enable us to monitor the fine scale phenology variability and also across the large spatial coverage as well. And the one possibility here we would like to highlight is probably associated with PlanetScope satellite. So PlanetScope satellite is operated by PlanetLab, a commercial satellite company from California in the United States, and is composite by more than 190 micro satellite. And this satellite nowadays have 3 meter spatial resolution with near daily temporal reverse cycles, and the data has a really good quality since 2018 and until now. So whether we can use this kind of like a technique to monitor the fine scale phenology, especially at the tree crown scale. To test this, here I would like to use a drone image we collect from the Panama Colorado Island as an example shown here. That's a drone image collect in the Panama forest. And we can carefully draw a polygon to segment each individual tree crown. Later on, we can also to calculate what's the crown diameter distribution for this forest site. And when we overlay with the different satellite with a spatial resolution, and we found really intriguing is PlanetScope with a 3 meter resolution almost can capture most of the big canopy tree crowns in the forest. This suggests this new satellite technique has the potential to enable us to study the fine scale phenology variability. And to test this idea, in 2019, my lab studied this kind of experiment. So we used the mountain Changbai canopy print site. That's a temperate deciduous forest in northern of China. So and in this forest site we have a canopy print tower, and in the tower we mounted like 3 pheno camera, so it can point to different directions. And on lab panel it just shows the zooming photos about different phenocane image we have there. And we also assemble the data from phenocane, and also we have some drone image, so then we can also monitor the fine scale of tree crown information as well, and also have the PlanetScope and also satellite data. So the main idea is try to use this example study site to test whether the novel remote sensing technique could enable us to monitor the fine scale phenology variability or not. And with the drone image, we can also carefully segment each individual tree crown, and together with the field survey, we can also identify each individual tree crown with different species and labeled as different colors. On the right panel, it suggests it's the enlarged photos from the drone image. That's how we segment each individual trees of different species as well. At the same time, we can use the PlanetScope satellite time series data, so then we can extract 2 phenology metric as shown here as an example. One is the start of leaf 4, and the other one is the end of leaf 4. These are 2 important metrics as an indicator of leaf 4 phenology. And we can use the PlanetScope satellite data to derive the phenology at the 3 meter pixel levels. And once we derive the phenology metric, we can overlay with the tree crown mask derived from the drone image. So then we can derive like a PlanetScope species levels phenology metric. And then we can also use the phenocane data installed from local site as a benchmark. And the results show as the bottom 2 panels. And overall, it suggests this kind of new technology, it seems it's possible to monitor at the crown scale or even a species level, a species scale of the phenology informations. So we found that's really quite exciting. So that's also a reason now we already started to expand it to more sites and to see whether this can be a consistent approach to monitor the phenology, especially at the crown scale. So we also really looking forward to any collaboration as well to see whether we can get more and more test this kind of idea in more and more region, and even we can start to build up more and more data set and also library as well. And like the last slide, I mentioned about the possibility to using the PlanetScope to monitor the fine scale phenology. But during the last 2 years, we also learned a lot when we deal with this kind of satellite data. And we found there are two important technical issues need to consider, several important technical issues need to consider when we process this kind of new satellite data. The first thing is PlanetScope satellite data is kind of almost similar as any satellite measurements. So as an ecology, we are really interested to understanding about the underlying biophysical processes. But at the same time, for any kind of satellite measurement, they will also subject to the artifact. And the artifact can be associated with a cloud and a shadow effect, and it can be associated with a sensor viewing geometry effect. And this is also commonly known as the bidirectional reflectance distribution function, or BRDF effect in remote sensing field. Another thing is the PlanetScope satellite is quite different from other satellites. For example, like the Maldives only have 2 sensors, but the PlanetScope satellite have more than 190 sensors. That means different sensors, they have, across different sensors, the DNA scaling also have inconsistency issues need to be considered. So that's the reason to address this kind of to minimize the artifact. And my postdoc, Dr. Jin Wang, during the last 2 years has been developing some technical advance. I think already we addressed most of these issues now. One is in order to address the sensor viewing geometry effect and also cross sensor DNA scaling inconsistency, and we come up a new approach to calibrate the PlanetScope data to a benchmark like a Maldives data. And then also recently we developed an automatic cloud and shadow screening approach so that it can facilitate this kind of data processing at a large spatial scale. So if you are interested to use PlanetScope data in your research, I will highly recommend to read these 2 papers. I think probably will provide some insight. And at the same time, we also have the code already developed. So we are happy to share the code as well. So the last part, now let us move to the last part. And last part, I primarily want to introduce how we can potentially using the vegetation spectroscopy technique to help to monitor the plant function trait. And the spectroscopy technique in the remote sensing field, we normally also call as the hyperspectral remote sensing. And the fundamental theory underlying this technique is based on all materials will interact with light energy in different and characteristic way, like the example shown on the left panels. And we found for the needle leaf and the broad leaf, their spectral reflectance actually are quite different. And the vegetation and also non-vegetation, they also have a significant spectral reflection difference as well. And also now with the more advanced in the remote sensing field, now we also have some like the spectral radiometer that can help us to characterize the light in narrow wavelengths reflected from or transmitted through plants and other materials. For example, like the data shown here, normally the data is now interpreted to the 1 nanometer resolution, but the sensor have the accuracy about 3 nanometers. So that's the reason we can now see a lot more detailed information based on hyperspectral reflectors. And then later on, we can develop some like models to infer key plant functional trait from hyperspectral data we collect. And like the example shown here, now we already have really high confidence to infer like a crop field from this kind of measurement. And we can predict leaf water content, leaf mass per area, leaf nitrogen, and so on. So many traits from this kind of technique. But if we talk about how to use the spectroscopy technology to infer like a leaf physiological trait like photosynthetic capacity, the pioneering work actually can be traced back to Dr. Shawn Serbin's this paper published in 2012. And the result actually are summarized in the middle panels. X-axis indicate is the spectral predict the photosynthetic capacity like this max, and the y-axis indicate is the likelihood derived this max. And overall, we can see the predict power actually are really strong. So that's the reason when the paper was published, it actually are quite exciting for the whole hyperspectral remote sensing field. But also there's some limitation about Shawn Serbin's work as well. The first thing is the original study was primarily focused on 2 temperate species. And then the model they are using is a static model running a process-based model. So after Shawn's work was published, later on, a lot of similar finding actually was found in different species, different sites, and even no matter in plants or in crop. But one central question remains in this field is whether this kind of approach can be generalizable across different species, different forest sites, and even at a seasonal time scale. So that remains unclear in the field for a long time. And that also raised a lot of doubt for people. They are not quite sure whether we should rely on this kind of technique to advance the plant functional trait monitoring, especially for the physiological trait. And why should we care about the generalizability issues? I think there are a lot of reasons, but I think probably to summarize, probably we can think about it from both methodology perspective and also from ecological perspective. Methodology perspective is if the model can be generalizable, that means once the model is fully developed, then all the community can be used, right? Alternatively, is if the model with more and more data are collected from the whole community, then that means we can make the model more and more applicable to other regions as well. And from ecological perspective, because this model rely on statistic model, if we can demonstrate this kind of model, there are large spatial or temporal scale convergence. That means that even is a statistic model, that still suggests there are fundamental, like some ecological constraint to link the spectrum and the trait, right? So I think that's the reason the generalizability issues is always a really important concern for this field. And during the past, I would like to address the generalizability issue from two perspectives. One is the temporal generalizability, and the other one is the spatial-wise. First, let us focus on the temporal part. On that panel, it summarizes, it just shows one example about the leaf scale monitoring we conduct in the Brazilian Amazon. Like we tag the leaf, then we monitor them from 10 days old until to 360 days old. At the same time, we also use a spectral radiometer to continue monitor their leaf aging processes. And like if we now just focus on panel A, if we just use our naked eye, we can already tell the morphology, leaf colors, and also probably like biochemical composition already changed with the leaf aging process. But the cool things about a spectral reflector is they provide a quantitative way to measure, to record this kind of information. So at the same time, we also measure a lot, like we not only measure the reflectance hyperspectral information, at the same time, we also use the LICO 6400 to measure the leaf level photosynthetic capacity as well. So then we try to see whether we can develop generalizable models to capture all multiple different dimensions, multiple sources of the variability of the VCMAX 25. The result, like the result shown here, X is a single spectral model predict of the VCMAX 25, and then different colors, including the data we collect from Panama and also Brazil, and also including the mature leaf and also young leaf. And overall, we found we actually can develop a generalizable model across all different kind of scenarios. This gives us a confidence that indeed have some like the seasonal convergence in spectrum and physiological trait relationship. And later on, we also want to use spectral to see whether we can use spectral to infer the leaf age, at the same time to use a spectral to predict the photosynthetic capacity, and see how whether by itself we can use a spectral reflectance, like time series of spectral reflectance information to help to check how photosynthetic capacity aging, right, change with the leaf age. The results are shown on the right panel, and the red color indicates is the field observations. The gray color indicates the spectrum model result. And overall, we found that using the spectrum model result, also it can help us to construct the lifetime change of the VCMAX 25, especially in the when leaf from new leaf to mature status. We found the model actually result are quite good, but definitely after the second phase from mature to old or deconsenescence, the model is not that well constrained, but primarily is because we have really limited data in all leaf age cohort. So that also suggests we need to collaborate with the whole community to see whether we can improve these kind of models in the next step. So later on, when I moved to Hong Kong U, so my postdoc, Dr. Tem Phenology and I discussed whether we can extend this kind of seasonal generalizability issues to explore spatial generalizability issues, so that we can go beyond the tropical forest to multiple different biomes as well. So we, my postdoc Tem Phenology led a team of students. So we did a intensive field campaign across the 3 different sites in mainland China, including Mountain Changbai, that's a temperate deciduous forest, then Mountain Dinghu, that's a subtropical evergreen forest, and then to Xianbana, that's a tropical rainforest. So we collect the data across kind of like a different biome to see whether there's like cross spatial generalizability issue or not. And here's your result. X-axis indicate is the spectral model predicted with max, and the y-axis again is the field derived with max. And overall, we found there's a strong cross-biome generalizability issues using the spectroscopy technique. At the same time, but also not quite surprising, is we also found at the same time using a spectral, it can help to infer many other important leaf traits as well. For example, like a leaf nitrogen, leaf mass per area, and leaf water content. And the follow-on, we also found really interesting things is we can use the spectroscopy approach to provide a novel way to represent trait-trait relationship. For example, now if we use the panel A as an example, x-axis indicate is the leaf nitrogen content, and the y-axis is the field the VC max 25 as an indicator of the leaf physiological trait. Gray colors indicate is the field observation. The red colors indicate is the spectral model prediction. And overall, we can tell the spectral predict the 2-dimensional scatter plot almost capture a quite similar scatter plot as the field observation. And also quite interesting is no matter whether the model or this kind of trait-trait relationship is weak or there's no relationship, using a spectral approach can capture this kind of like a relationship quite well. And why this is important, like Jing Yang pointed out, for the next generation of our seasonal model, we are soon move on to a trait-based ecosystem model. And a lot of trait-based models sometimes using a trait covariance. Even in the real data, it shows there's a weak relationship or no relationship. But in the models, sometimes we actually use a linear relationship, and then with the assumption is they actually are highly correlated. But if we can use a spectral approach to do it, that means we doesn't need to depend on any prior assumption whether this kind of trait relationship have type relationship or doesn't. So that's the reason I feel this can be another things quite important for the next generation of the trait-based modeling as well. So here is a quick summary about what we just discussed here. The fourth thing is I just show you we just talk about leaf phenology is tightly linked to plant photosynthesis seasonality, and there are at least 2 aspects of leaf phenology, like quantity and quality aspect. And later on, I show you there are potential 2 important remote sensing technology can help us to quantify the fine scale phenology variability, including high spatial resolution. Seasonalit can be a way to help us to monitor fine scale phenology probably at a count scale. And this is also quite important for us to, in the next step, to understanding about the individual to ecosystem phenological scaling process as well. And also hyperspectral remote sensing, or we call vegetation spectroscopy technology, can also offer a really effective way and also scalable means to help monitor a spatial and temporal variability in leaf physiological trait. So, and with this, I also would like to acknowledge my postdoc and also my student and also my collaborator to support on this work, and also my funding source from both mainland China and also from the Hong Kong. And also thank everyone for your listening. And also, if you are interested, then you can visit my lab webpage, and that's the main page of my lab web. So we are develop a lot like a multi-scale approach, try to advance multi-scale remote sensing approach, try to advance the link carbon cycling questions. Thank you again to everyone. I'm happy to take any questions if time is allowed.